This work we published in 1990, and one of the authors on the paper was Professor Pauling, the same person who had encouraged us to look at the effects of nutrients on vitamin C. And we published it in a very prestigious journal, the National Academy of Sciences, the United States. It's been 18 years. And you can wonder why, when such a result was already published 18 years ago, that mainstream medicine has not yet woken up to the implications of this research. That itself tells you that they have a certain agenda of promoting their own types of drugs in order to treat disease. In this same research, we also found that if you looked at, so the result before was with cells that were freshly infected, just newly infected cells. Then we used cells that were chronically infected, that are continuously producing virus, the same type of result for a longer period of time. By day 4, the level of virus was down to less than 1% in the culture medium of the infected cells. Very dramatic effect of vitamin C. This compares vitamin C with a known drug called AZT. Actually, what is shown here is that you can also see this effect in cells that are harboring virus, called latent infection. As you know, in HIV infection, when a person gets infected, very early on there's acute infection. For a few weeks the virus can be detected in the blood, and the virus goes into hiding. That's called clinical latency. And it can be latent for many, many years until it gets reactivated. So we know that there are what are called latently infected cells, cells that have the virus, nucleic acid, the genome, but they are not actively producing virus. That's what's shown here. This is the control, latently infected cell. But you can induce them to produce virus. This can be done in the laboratory by adding certain agents. TNF-alpha is like a cytokine, a product of the immune system, inflammatory cytokine. So when the level of inflammatory cytokines go up, the virus that is in hiding comes out. You get high levels of production of the virus. Now in this system, if you add in vitamin C, these are different levels of vitamin C, the AA is ascorbate, it shows you that this simple vitamin can suppress this activation of the virus from latently infected cells. If you compare this with the known drug AZT, and that's what's shown here, you find that AZT has very little effect in this system. Meaning that if there is latent infection, an antiretroviral drug such as AZT is not necessarily going to be effective in suppressing its activation. And this activation can occur when an individual is exposed to stress or to any kind of changes. There may be hormonal changes that occur in the body, and the virus comes out of these latent cells from hiding. That in this system, if you compare what a nutrient like vitamin C does to a known antiretroviral drug, you can see the differences, that one is very active but the other is not. So this is actually a summary of the amount of changes. There's about anywhere from 2 to 10-fold reduction with vitamin C, less than 2-fold with AZT. This is also a study that we did in infected cells that we published several years ago. Another nutrient we talked about earlier is the amino acid cysteine. Cysteine is very important in AIDS. First of all, it's an essential amino acid, And we know that there is cysteine deficiency in AIDS. I was talking earlier about glutathione, and I mentioned it's a tripeptide. One of the amino acids is glutathione and cysteine. If you measure in the blood of infected people, you'll find that there is cysteine deficiency. The blood levels are lower. But cysteine by itself is not recommended to be taken as a supplement. Not well absorbed, it can have toxic effects. So, in place of cysteine, a derivative of cysteine called N-acetylcysteine, or NAC, is used. NAC has some powerful effects in the body. For example, it's an antioxidant, antiviral agent. And there were studies done at a university in the USA called Stanford University, where N-acetylcysteine was given to people with HIV infection. It was found to improve survival in these patients. Just supplementing with NAC increased the survival of people who are HIV infected. Our own work that we have conducted has shown that NAC, like vitamin C, can also suppress HIV in infected cells. And this slide is to illustrate that if you combine two nutrients, if you combine vitamin C and NAC at very low to moderate doses, because at low to moderate doses, this is the control level of virus, and these are low to moderate doses of these two nutrients, they have some effect on suppressing virus, but not substantial effect. However, if you combine the two, then you get a cooperative effect. The two together are working better than either agent alone. This is the principle behind combinatorial nutritional therapy. Why, for example, we recommend or suggest that a nutrient combination be used. It is because of this type of work that when you combine these nutrients at very low levels, they have like a cooperative effect on stopping virus production. Now, some other more recent results with these specific nutrients, and I'm talking about some of these compounds because they are present in the nutritional supplement that we are distributing through Sanco. These ingredients that are there in the nutritional supplement have been researched. This is another ingredient, green tea extract, an extract from the leaf of green tea, also an antiviral agent. Shows here that in chronically infected cells, as you increase the dose, you get suppression of HIV. And if you use a nutritional mixture that contains green tea extract, vitamin C, N-acetylcysteine, then the nutritional mixture beyond certain doses also reduces the level of virus in these infected cells. The effect that was seen with individual nutrients now can be reproduced with a combination of nutrients. We are what we call a synergistic effect. When you combine the nutrients, the effect of that is greater than the single nutrients alone.